CBSE Class 12 Practical Skills in Biology. Experiment number 3 Water. 3.1 To study the pH of different water bodies. Aim: to study the pH of different water bodies. Materials required: water samples from borewell, hand pump, pond and sewage. Test tubes, universal pH indicator solution, pH indicator paper, narrow range and broad range. Procedure: pH is an indicator of acidity or alkalinity of a sample. Pure water has a pH of 7, which indicates neutral nature of water. Neutral pH is preferred for growth of most plants. There are different methods to determine the pH of a water sample. Water is an important factor that affects the growth of all living organisms. Using universal indicator solution, take water samples from different water bodies like borewell, hand pump, pond and sewage in test tubes and label them as A, B, C respectively. Now add 5 drops of universal pH indicator solution to the test tubes A, B and C. Note down the color developed and compare it with the color chart of the universal pH indicator solution for each test tube. Using broad range pH indicator paper, take water samples from different water bodies like borewell, hand pump, pond and sewage in different test tubes and label them as A, B, C respectively. Now take a small strip of broad range pH indicator paper, that is pH 2 to 11, and dip it in test tube A. Note the color developed and compare with the color chart given on the broad range indicator paper and get a rough estimate of pH of the sample water body. Repeat the steps 2 and 3 for water samples in the test tube B, C and D. Using narrow range pH indicator paper, take water samples from different water bodies like borewell, hand pump, pond or sewage in test tube and label them as A, B, C. Now take a small strip of a suitable narrow range pH indicator paper. For example, if the pH of water sample is determined as 8, choose a narrow range 7 to 9 and dip it in a test tube A. Note the color developed and determine the pH to the nearest possible value with the help of the color chart. Repeat the steps 2 and 3 for determining the pH of samples B, C and D. Observations: Result: To study the clarity of water samples. Aim: to study the clarity of water samples. Materials required: CC's disc, rope, pins, meter tape. Procedure: pure water is neutral, tasteless, odorless and has dissolved nutrients and gases like O2 and CO2. It is not turbid, that is, it is a clear solution. Turbidity of the water body determines the depth up to which light can penetrate and hence affects the distribution and photosynthesis of phytoplanktons and microphytes. Reach the center of a nearby pond in a small boat. Now slowly immerse the CC's disc into water, holding the rope tightly till the black and white segment of the disc disappears. On reaching of particular depth, the disc becomes completely invisible. Now mark the length of the rope with the help of a pin when the disc disappears, say X centimeter. Now slowly pull up the disc till the black and white segment of the disc just reappear. Mark the length of the rope where they just reappear with the help of a pin, say y centimeter. Find the lengths x and y using a meter tape. Find the mean length, say z centimeter of the rope by using the given formula z is equal to x plus y by 2 centimeter. Observations. Results. The value Z represents the depth of the floating zone up to which sunlight penetrates in a water body and photosynthesis takes place. The water sample from crystal clear deep lake has high value of Z and thus has high clarity in water. The water sample from stagnant water bodies like lake has low value of Z and thus the water sample is turbid. 3.3. To analyze living organisms in different water samples. Aim: to analyze living organisms in different water samples. Materials required: water samples from different water bodies like lake, pond, river, etc. Beakers, small test tubes, slides and cover slips, watch glasses, dropper, compound microscope, 5% FAA, that is formalin acetoalcohol, in a ratio of 5:5:90, and formalin to acetic acid to ethanol as preservative. Procedure: collect about a liter of water sample from each of the different water bodies like lake, pond, river, etc. Now add about 5 ml of FAA to each of the different water samples to fix and preserve the living organisms present in each sample. Label each of these cylinders to indicate the site from where these water samples have been collected. Decant off the clear water, leaving concentrated sediment at the bottom of each cylinder. Transfer the sediment of each cylinder into small test tubes. Now transfer a few drops of sediment liquid from a test tube into a watch glass using a dropper. Dilute the sediment with water if the sediment is highly concentrated. With the help of a dropper, transfer a drop of this sample from the watch glass on the center of the slide and mount it with a cover slip. Remove the excess water using blotting paper. Place this glass slide on the stage of compound microscope. Prepare a few more slides and each water sample in the same way. View each slide first under lower magnification and then under higher magnification. Observations. Record the different types of organisms present in each water sample. Count the number of organisms under each field of microscope. Bacteria. Different types of bacteria have different shapes such as spherical, rod-like or spiral-shaped. These are microscopic unicellular, without nucleus, lack of membrane-bound organelles. For example, Koki, Bacilli, Vibrio, Spirulli. Protozoan. These include unicellular and uninucleate zooplanktons. For example, amoeba, paramecium, Euglena, Actinophores, Giardia lamblia, Opulina. Algae. These are plants which can be either unicellular or multicellular. They grow abundantly in water bodies and are producers of aquatic ecosystem. For example, Chlamydomonas, Volvox, Spirogyra, Oscillatoria, and etc. Larvae and nymphs of arthropods. Larval and nymph stages of various insects are found in most of the water bodies. Few of them are shown in the figures below. Amphipod, mosquito larva, isopods, Disticus larva, Disticus, water scorpion, first instar mayfly nymph, second instar mayfly nymph, instar nymphs of dragonfly. Result: samples collected from different water bodies show variations in type and density of different organisms.